Hello, my name is Ali, and this is my presentation for my research on the effect of lead on metabotropic glutamatergic receptors in the brain. I'll begin by talking about the environmental toxin that my project focuses on, which is lead. Lead is a naturally occurring toxic metal that is exposed through mining, manufacturing, leaded paints, and lead acid batteries for our cars. It can also be easily exposed through household items such as toys, jewelry, cosmetics, and drinking water. Chronic exposure to lead has the potential to impair cognitive development, particularly in the developing brain. This poses a severe threat since children who have lead poisoning have a really high risk of developing cognitive deficit. Learning and cognition depend on certain mechanisms such as neuroplasticity, which is the ability of the brain to change and adapt during one's lifetime. Mechanisms like these depend on the transmission of the neurotransmitter glutamate. Lead, however, specifically targets glutamate receptors in the brain. My project aims to answer how exactly lead impacts these receptors in the brain, hoping to shed light on how it affects cognitive function. The specific receptor my research focuses on is the mGlu-R7A metabotropic glutamatergic receptor, which is found in high concentrations in the hippocampus. The function of this receptor can be modeled using the G-protein coupled inwardly rectifying potassium channel known as GERK. This channel is activated by the mGlu-R7A receptor, so its response to lead is also tested as an indicator of mGlu-R7A function. This was done in a previous experiment by recording the response of Xenopus laevis oocytes to lead. Xenopus laevis is a species of frog that are great for electrophysiology and contain the same mGlu-R7A receptor. The two electrode voltage clamp method was to be used for recording current from the mGlu-R7A receptor and GERK in the oocytes. In a previous study done, 100 micromolars of glutamate was applied to the oocytes and the current was recorded. The concentration of lead varied, so as you can see in figure 1 right here. This is a graph of raw data produced during previous experimentation done on the effects of lead on this receptor. The x-axis is time and the y-axis is the current. So on the x-axis you can see the 2-minute interval where 100 micromolars of glutamate was applied. You can also see the variation in lead concentration from all the way down to 0 to up to 100 micromolars. So as you can see in the shape of this trace, the upward deflection is indicative of the inhibition of the inward current. The downward dips actually mean activation, so the fact that there's upward inflections indicates that there's inhibition. Now the inward current occurs when potassium flows through the GERK channel from the outside to the inside. GERK is responsive to G-protein subunits that respond to mGlu-R7A and is a great readout for this G-protein coupled receptor. At higher and higher concentrations of lead, data shows that there is increased inhibition of the mGlu-R7A activated GERK current. Now in the second graph from this previous study labeled figure 2, it supports the hypothesis that the mGlu-R7A receptor is indeed what is causing GERK to behave this way. The x-axis shows the lead concentration in micromolars and the y-axis shows the percent maximal response. You can see that in this figure, when lead is applied to GERK coupled with mGlu-R7A, there's a significant amount of inhibition, especially at higher concentrations. Furthermore, when lead is applied solely to GERK, as shown by the red dots, there's also inhibition occurring at higher concentrations. This data supports the hypothesis that lead is indeed affecting mGlu-R7A, which in response is affecting GERK. Now all of this data is statistically relevant because these concentrations of lead used in the experiment are an accurate representation of levels of lead that are involved in lead poisoning. This supports the hypothesis that lead has the ability to impair function of this receptor at physiologically relevant concentrations, and therefore may impair cognition in individuals with lead poisoning. And that concludes my presentation. Thank you.